It is then now my pleasure to introduce our keynote speaker, Professor Stuart Cole. So I'm very disappointed that he's not here in person, but it is exciting for me to hear about his work. He's an officer of the Légion d'honneur in France, as well as a fellow of the Royal Society. And I have to admit, I had to look this up, he's a Knight Commander of the most distinguished Order of St. Michael's and St. George. I think that is a very prestigious honor, particularly for a scientist. But as a scientist, Professor Cole is an internationally renowned microbiologist working in the global health field for his entire career. With over 350 different scientific publications, he's really made groundbreaking contributions in HIV, TB, and HPV genomics, and in AMR research. Most notably, his work on the pathogenicity, evolution, and genomics of pathogenic mycobacteria, particularly those causing tuberculosis and leprosy, has accelerated TB drug and vaccine discovery and development. And in fact, I was pleasantly surprised to find out about all of the numerous patents that he has been an inventor of, and that has led subsequently to diagnostic tests and candidate drugs, several of which are currently in clinical trials. Over the course of his career, he has previously led the Global Health Institute at the Swiss Federal Institute of Technology in Lausanne. And then he was a past president of the Institut Pasteur in Paris from 2018 until December 2023. And since then, he has joined the INEOS Oxford Institute for Antimicrobial Resistance as its executive chair. So with that, I'd like to welcome Dr. Professor Cole.
Well, I'm very sorry that I'm unable to be there with you today, but I had some health problems as a result of viruses and bacteria ganging up on me. It was a great disappointment for me not to be able to visit McGill and Montreal, which is a city that I appreciate. I was greatly looking forward to my trip, but it wasn't to be. So I shall do my best by Zoom, and I hope that what I say will be of interest and relevance to your own research. So let's start sharing. So as I was saying, I currently work at the INEOS Oxford Institute for Antimicrobial Research, and this is an institute which was set up by the University of Oxford thanks to a major gift from INEOS, which is a petrochemicals company. And the owners of INEOS were particularly concerned about the rise in antimicrobial resistance. And so they wanted to use their philanthropy to try and make a difference. And so today I will tell you something about the work which we've been doing at IOI, as we call it. And also I'll talk about AMR in general. So antimicrobial resistance or antibacterial resistance. And we all know that this is one of the major threats to global public health, and that if we don't take radical action, our current way of life will be severely impacted. And AMR is sometimes referred to as the hidden pandemic. So I'm going to start with a brief introduction to a little bit antibiotics, and then I'll talk some science. Then we talk about how AMR rises, the scale of the problem, the difficulties in producing new antimicrobials, and I'll then tell you about some successes coming from the tuberculosis field, and I'll end by showing how you can help. And it's particularly apposite that I should give this talk this particular week because it's Antibiotic Awareness Week. 1 year ago at the United Nations, there was a high-level meeting held on AMR at the 79th General Assembly. And the various country experts got together to share their plans and their ambitions for trying to make an impact on the AMR problem. And you can see here the can you see my mouse or not?
Yes. Yes.
Yeah. Okay, so these are let's see if I can turn the laser on. Can you see the laser?
Yes.
Okay, so this is the summary statement which arose from that meeting, the executive statement from the 4 UN bodies, FAO, UNEP, WHO, and OAH, and they set themselves the objective of reducing the number of human deaths from AMR by 10% by the year 2030. So that's to say a reduction in mortality due to AMR of between 1 and 200,000 per year. So I thought this was a rather modest objective, but it does have the merits of being attainable. So a few words about antibiotics. We all know that antibiotics, the term is from the Greek agent against life, antibios, and that the antibiotics are currently the foundation of modern medicine as we practice it. Initially, antibiotics were used to treat bacterial infections, but they've subsequently found much broader application and have made medical procedures such as surgery, cancer therapy, and organ transplants possible by reducing the risk of infection. And it's somewhat ironic to note that it was the discovery of antibiotics, which I think played a major role in launching the modern pharmaceutical industry. And it's now almost 100 years since the first antibiotic was discovered. And this was, of course, Fleming's discovery of penicillin, of the Penicillium notatum mold in 1928, which was shown to produce an inhibitory substance that was subsequently found to be penicillin, and this was purified in Oxford and tested on some small group of patients and shown to be efficacious. In the Second World War, the scale-up of production of penicillin was massively improved, and the antibiotic then became widely used. And in the following decades, many more antibiotics were also discovered. There have been relatively few new antibiotics, and the last major class was discovered in the 1980s. In the 2000s, we became aware of the growing problem of antibiotics and how this not only menaced our existing treatment options, but was undermining the foundation of modern medicine. So this is an old slide which just shows you the timeline of the clinical use of the antibiotic, followed by when resistance was identified. And you'll see that for all the 4 classes shown here, penicillins, tetracyclines, macrolides, and quinolones, that within 3 or 4 years, there were problems of antibiotic resistance detected. Now, Fleming himself noted, and he made this cautionary statement, that there may be a danger, though, in underdosage. It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentrations not sufficient to kill them, and the same thing has occasionally happened in the body. So Fleming was certainly ahead of his time in that respect. And the antibiotic that interested him, penicillin, we know now that resistance to this is mediated by an enzyme, a beta-lactamase. And the beta-lactamases are a very widespread source of resistant to beta-lactam antibiotics. These can include the meropenem, one of the last generation of antibiotics, but also cephalosporins and penicillins. So the enzyme binds the meropenem, and then there's a serine hydrolase reaction which cleaves the beta-lactam bond, resulting an inactive derivative and the complete loss of antibiotic activity. Now, the enzymes responsible for this are often encoded by plasmids, and these plasmids are transferable among many different groups of bacteria, including the so-called escape pathogens, which are the ones which are of greatest concern in human medicine. So this is the E. coli, staph, Klebsiella, Acinetobacter, Pseudomonas, and Enterococcus. And the transfer of these resistance genes is underpinning the widespread nature of AMR nowadays. So if we look to see how and where resistance occurs, it stems from the overuse of antibiotics, but sometimes even the inappropriate use. In certain settings, in low and middle income countries, for instance, the antibiotics are available or are of poor quality, maybe counterfeit, and not have the full concentration of the active pharmaceutical ingredient. The main drivers are the intensive antibiotic use settings, so hospitals and the agriculture sector. And I'm going to say a few words about agriculture next. So this slide shows you the relative use of antimicrobials, so in human medicine here, and shall we say in agriculture. And there are somewhere in the region of fourfold more antibiotics used in the agricultural sector than there are in human medicine. And one of the reasons for this is due to the increased need for food to feed the growing human population, in particular sources of protein such as poultry. And it's been predicted that in the next 25 years, the number of poultry in the world will increase 2 to 3-fold. And this will also be accompanied by increased consumption of antibiotics. And the antibiotics are currently used a lot in intensive farming settings where infections may be common. The main use of the antibiotics in agriculture is an antimicrobial growth promoter. So it's not a health argument, it's an economic one, bringing the meat product to market and a shorter timeline. So it's estimated that this can reduce the time to market by a factor of 15%, which I think is rather modest and unacceptable. The use of antibiotics has been banned in agriculture in the European Union and UK, and I was pleased to see that Canada is fully aligned with that policy. And one reason why I think this is not a good way forward is that there are far more acceptable alternatives which are available. And I would like to illustrate that by showing you an example from Norway. And Norway is a major producer of fish, notably farmed salmon. And this slide was kindly given to me by Dr. Cahoon from the Norwegian Veterinary Association, who produce a report every year showing the amount of fish which is produced as a function of the consumption of antibacterials in the fish industry. And so here we have a 40-year timeline starting in 1981, and you see the blue bars correspond to the tons of antibacterial agents which have been sold. The green curve is the biomass of farmed fish. And you can see there's been an enormous increase in output in that 40-year time period, concomitant with a huge decrease in the use of antibiotics. And the reason for that was in the 1980s, the Norwegians started introducing vaccines to protect farmed fish against infection. They then found that these could be improved by adjuvanting with different oils. And in the subsequent years, they worked on a multi-component vaccine which protects against 7 different fish pathogens. And this, I think, is a brilliant example of how you can remove the antibacterials from the agriculture sector through the intelligent use of vaccines. And this is something which I think would be great to see more widely used. So I'm now going to turn to the human situation, and I'm going to say a few words about the global AMR burden. And this was the data for 2024 taken from this major publication, which came out to coincide with a high-level meeting at the UN last year, a Lancet paper by what's often referred to as the Graham Group. So the global burden of bacterial antimicrobial resistance, 1990-2021, a systematic analysis with forecasts to 2050. And this paper had a big impact and was taken up by the press around the world, including one of your local newspapers, Le Journal de Montréal, which flagged up the potential danger caused by AMR. If we look at this in a little bit more detail, the burden concerns all classes of antibiotic. It affects all countries, but the disease burden is worse in the low and middle income countries. The Graham Group estimate that somewhere in the region of 40 million lives will be lost by 2050 due to AMR, and this can be broken down into 2 forms. Loss of life directly due to an antimicrobial resistant pathogen, so in sepsis, for instance, or indirectly or associated with AMR, where there's a much higher number. So this could, for instance, be cancer patients who subsequently become infected with an AMR pathogen, and the cause of death is attributed to cancer rather than AMR, of course. Now, the Graham report predicts that better hygiene, healthcare, vaccination, and a new antibiotic for gram-negative infections could save 92 million lives by 2050. And I would now like to just dive into some of the demographics presented in that paper. And this shows you the number of deaths associated and attributable as a function of the year. So in green, we have 1990, and in dark blue, 2021. And then we have the different age groups here, from neonates through to individuals over 95 years old. And so what you can see is that in 1990, the peak of the deaths was in the under 5 age group. Whereas now in 2021, the peak has shifted to the elderly, so there's been a big change in demographics. So from this, we conclude 2 things. First of all, I think that age matters, so the elderly are more prone to AMR infections. And secondly, I would like to propose that the widespread rollout of the multivalent pneumococcal vaccine has contributed to preventing deaths in the under 5 age group, again showing the potential benefit of vaccination. Just coming now to some geography, the global distribution of the AMR burden, color-coded according to the mortality rate. The worst affected countries in 2021 were shown to be in the African continent and some parts of Asia. But the modeling suggests that there will be a shift in the trend with a big increase in AMR in South America, in India and the Indian subcontinent, other parts of Southeast Asia, and also in the Russian Federation. So this is clearly a very alarming modeling data, and we need to take some action to try and make sure that this does not occur. One of the most important statements in the Graham report I've summarized here. It encapsulates everything. Given the high variability of AMR burden by location and age, it is important that interventions combine infection prevention, vaccination, minimization of inappropriate antibiotic use in farming and humans, and research into new antibiotics to mitigate the number of AMR deaths that forecasted for 2050. So I put in bold here research into new antibiotics, because now I would like to introduce you to the, shall we say, the new antibiotic pipeline. From these two publications, I concluded that in 2022, there were 26 new antibiotics in clinical trials. However, only 4 of these were actually new chemical entities. The bulk of them were next generation beta-lactams or cephalosporins and so on, or beta-lactamase inhibitor. There was a lack of, which we say, innovation. If we look at this as a function of the total number of drugs which were approved in this 40-year window here, you can see that very clearly there's been an increase in new drug approvals in the recent time. In this column, you see that 226 new drugs were approved for various interventions in human medicine, whereas there are only 5 antibiotics. And this is a staggeringly depressing situation. One of the reasons for this was identified by the Wellcome Trust when they performed an analysis of the antibiotic market, and they found that in that time frame, the number of new antibiotics being approved was declining, as I've just indicated, but in parallel, the number of global pharmaceutical companies active in the area has gone down from 18 to 5. So another warning sign. Let's look at the sales now from the top 200 medicines report for 2022. And in that year, the best-selling drug, if you like, was the COVID vaccine, the Pfizer-BioNTech one. And the 2 other the next second and third best-selling drugs were Keytruda, a monoclonal antibody widely used in cancer treatment, and Humira, which is a monoclonal antibody used to treat rheumatoid arthritis. In the top 200 best-selling medicines, there was only 1 anti-infective, and that was the pneumococcal vaccine, which I alluded to a few moments ago. So why are we in this alarming situation? Well, the trend indicates that big pharma is almost gone. And if we look at the biotech sector, we see that it's severely underfunded. So a few bullets, pharma does actually very little R&D in the antibiotic arena. They rely on acquisitions to fill their pipeline, so that this means that most of the de-risking, the front end, the discovery, through early stage preclinical development is done in academia and the biotechs. The biotechs are struggling, and I've given you 2 examples here, Achaogen and Melinta, Both of these filed for bankruptcy after bringing new antibiotics to market. So these are approved antibiotics. And the reason for the bankruptcy was that they brought in too little revenue. So Achaogen had an injectable aminoglycoside used for complicated UTI, and Melinta had a couple of interesting drugs, a quinolone and an oxazolidinone, but that was insufficient for them to generate any revenue to sustain the company. So from this, we can conclude that the main negative factor is economics. It's costly to develop an antibiotic, and failure in clinical trials is relatively high. In addition, new drugs are in competition with the currently existing antibiotics, which can be produced in ton scale at an inexpensive rate. Furthermore, antibiotics are used sparingly to protect them, so they're often referred to as last resort medicine. And the window of use is very short compared to chronic diseases. So in turn, this means that the profits are meager compared to what happens in the cardiovascular disease sector, in cancer, diabetes, and so on. But nonetheless, the societal value of antibiotics is enormous. And so something has to be done to reverse this sad situation. So a couple of initiatives have been proposed, and these are referred to as push and pull initiatives. In the push, this involves funding early stage work, so on preclinical and early clinical development of new antibacterials. And so examples of that could be, for instance, the INEOS Oxford Institute. In the TB field is what the Bill and Melinda Gates Foundation have set up, the TB Drug Accelerator. And then there's quite a few NGOs, such as CarbX from Boston, GuardP, a spin-off from Drugs for Neglected Diseases Initiative. And then in the European Union, we have what's known as the AMR Accelerator. Now, these initiatives often involve public-private partnerships, so combining the agility and innovation of academia with the pragmatism and know-how of industry. However, late-stage clinical development is the most expensive part of the whole process, and this is where pull initiatives are required. And there are a couple of things which have been proposed. In the United States, the FDA has what's known as a priority review voucher, and these are available for a number of therapeutic areas, pediatrics, medical countermeasures, and so on. And what a PRV means is that if a company brings a product to market, they're awarded a PRV, which can then be either used to accelerate review of their next product by FDA, or, and this is happening increasingly, these PRVs can be sold to another pharma company. In 2017, the UK government commissioned Jim O'Neill to investigate the economics of the AMR field, and his team produced this report tackling drug-resistant infections globally. And one of the things that the O'Neill report suggests is the implementation of something akin to the Netflix model, so whereby you take a subscription and then you can use as much or as little antibiotic as you need. Another proposal that they came up with is what's known as market entry rewards, and these are lump sum payments when a drug makes it to market. So this figure shows you a schematic of what this is supposed to look like, although it's not been implemented. So the costs are shown here as those incurred by the healthcare sector, the payer, or by the drug developer down here at the bottom. And you can see that in the first years of antibiotic or drug development, all the costs are borne by the drug developer. And it's only when the drug is registered and gets onto the market that there is the possibility of raising money from sales. But as we saw with the examples of Achaogen, for instance, and Melinta, these may be insufficient to offset the initial investment. And this is where the market entry reward comes in. So that these are the orange blocks here. So these would be lump sum payments for a specific period of time until the revenues are sufficient to sustain the drug or the company with the biotech producing it. Now, these are all examples from the northern hemisphere, the Western world, how pharma works there. And I'm inclined to think that maybe some of the developing countries in the BRICS could actually do this better. And I'm particularly encouraged by some of the signs I'm seeing from India, Indonesia, and of course China. So perhaps they'll come up with something more creative and more cost-effective than the Western models. I'm now going to change gears and talk to you a little bit about tuberculosis. And somewhat unusually in today's climate, I would like to pay tribute to a president of the United States, and this is George W. Bush. George Bush commissioned Tony Fauci to set up something known as PEPFAR, the President's Emergency Program for AIDS Relief, because he was struck by the lack of access to antiretrovirals of many HIV/AIDS individuals during one of his visits to Africa. And he felt that we had the drugs there, and it was important to make them available to people in need. And this is what PEPFAR did. It had a knock-on effect in the field of TB, because TB and HIV often co-occur. The two diseases drive each other, and consequently there was more investment into the TB sector. So many thanks to George W. Bush for launching this initiative. It saddens me to see what's happened subsequently. Now, if we look at the situation in TB, there's been a big improvement in the past 30 years. 1994, the incidence was somewhere in the region of 15 million new cases and 3 million deaths. 2024, the number has come down in terms of incidence by a third, and as have the number of deaths. And this is in the context of a bigger global population, of course. So we've made an impact. Prevention is still inadequate because there's too many cases occurring, new cases, but the outcome is considerably better. There are far fewer lives lost to tuberculosis. And the reasons for this are better rollout and a better drug supply. And this is in part thanks to initiatives like PEPFAR, but also the contribution of the Global Fund and the World Bank, etc., which have made drugs, treatments more accessible to patients in need. The increase in the R&D linked to TB has also resulted in us having better perspectives for future treatments. And we now have improved diagnostics, drugs, and vaccines. And much of this has been funded by the public sector, by the European Union, NIH, and also by NGOs such as the Gates Foundation and so on. And we must thank them for their support of these important interventions. I'd just like to show you the TB drug pipeline here in 2025. So it's broken down into the different phases: discovery, preclinical development, and clinical development. And if we just look at clinical development, we have the phase 1, phase 2, phase 3. And in TB, there are currently 21 new drug candidates in clinical trials. 3 new drugs, bedaquiline, delamanid, and pretomanid, were approved a few years ago. And I'd like you to remember the situation with the AMR field in general. So here we have as many candidates in clinical trials for TB as for the whole of the AMR sector. And in addition, there are 4 times as many new chemical entities in development for tuberculosis than there are in the AMR for new drugs for escape pathogens, for instance. So I think that's a pretty remarkable achievement. And this, I think, is stunningly exemplified by the availability of a much simpler, faster treatment for multi-drug resistant and extensively drug-resistant TB. And this was shown in a clinical trial known as NIX-TB, promoted by the TB Alliance. In this hand, you can see one day of a typical XDR TB treatment, and on the other hand, you can see the new regimen, which involves far fewer drugs and a duration of 6 as opposed to 18 months. And so this new regimen has been referred to as BPAL. It contains 3 drugs: bedaquiline, pretomanid, and linezolid. And there are improvements on BPAL currently in progress. So I think this is a spectacular example of how dedicated drug discovery and drug development can have a huge therapeutic impact. Bedaquiline was discovered by Johnson & Johnson, and somewhere in the region of 0.8 million MDR-TB patients have been treated with the drug now, and mostly cured. And in the process, somewhere in the region of 6 million new infections have been averted, according to the modelers. So I think we should pay tribute to Johnson & Johnson for this pioneering work with bedaquiline, this game-changing new drug which inhibits the ATP synthase of Mycobacterium tuberculosis. Sadly, despite the success of bedaquiline, Johnson & Johnson have recently exited the infectious disease R&D space, and this pains me greatly. However, there are a number of other initiatives ongoing in the world, and this is one with which where I'm the academic leader, and this is what's something known as ERA40B, or the European Regimen Accelerator for Tuberculosis. So this is a push initiative with funding made available by the European Commission as part of its Innovative Medicines Initiative, and with matching funding from the European Federation of Pharmaceutical Industries and associated partners. So we are 31 full members in ERA40B, 22 from academia, 3 small medium enterprises, and 6 from the pharma sector. You can see the geographic distribution here with collaborators in the United States. The budget is indicated there to 207 million for this 6-year program. And the aim of the European Regimen Accelerator for tuberculosis is to take new candidates through early and late preclinical development and through to the end of phase 1 clinical trials. At that point, they're transferred to another push initiative, if you like, this one known as Unite for TB, also funded by the Innovative Medicines Initiative. This has 30 partners, 4 pharma companies, and 13 different countries involved. The budget is a bit smaller. Among the many assets which they have are 40 clinical trial sites located on 4 different continents. And of course, they have a range of candidate drugs coming from ERA40B and other initiatives such as the TB Drug Accelerator. So this program is led by the Helmholtz Munich, and the main partners are Radboud University, GSK, and Oxford University Clinical Research Unit. So that's the management team. Again, funding from the European Commission under the IMI. So I would like to state that I think 25 years of effort in the TB treatment field has brought its reward. But TB in some ways is much simpler than AMR, which in itself is a vast problem with multiple components. TB is a chronic disease. It's not an acute disease, so it's in a way simpler to treat. There's also there's less variation in terms of the pathology, since it's primarily a pulmonary disease, unlike different AMR infections. One of the reasons for the success in the TB field is that despite initially work in silos, the community has been brought together to work as one rather than a bunch of disconnected individuals. And this was made available by various funding schemes such as those that I've just introduced you to. As a result of that, we have much better cohesion in the TB drug development space. There are fewer gaps in funding, so things can move forward faster. And I think one of the reasons behind the success is that in tuberculosis, there's no profit motive. It's hard to make a profit in AMR generally, but tuberculosis affects primarily the low and middle income countries. So profit margins, if they existed, would be very, very modest. And I think this removes the competitiveness which one often sees in the pharma sector, and results in a healthier and more open working environment. So what can we learn from what's been done with TB to tackle the AMR problem in generally? So here I put up a number of what I consider to be obvious and important points. So starting with diagnostics, I think we need to be much smarter in terms of using diagnostics, not just for identifying the pathogen involved, but also for rapid drug susceptibility testing. So this will enable us to better match the treatment to the pathogen, so less inappropriate use of antibiotics. TB relies on combination therapy. This is something which is rarely done in the AMR field for a variety of reasons. It's something which I feel merits further investigation. However, to obtain the paradigm shift which we've seen in the TB field, it's important to relaunch discovery efforts to find new antimicrobials. And here I feel that rather than persisting with the classical approach that pharma developed so brilliantly in the 60s, looking for broad spectrum antibiotics, that it might be better to, should we say, lower the bar and target pathogens on an individual or a small group basis. So, for instance, try and find a new intervention to treat multi-drug resistant infections due to Klebsiella pneumoniae. Obviously, we have these amazing new tools that have come from the IT sector, so machine learning and artificial intelligence, and these are underpinning more and more drug discovery efforts. But all of this will fall apart unless we have a mechanism to sustain funding of the discovery and development efforts. So I'm nearing the end and my voice is weakening, but I'm going to sum things up with 2 slides. First of all, I would like to show how you can help reduce the AMR burden. So obviously the first one, this is a no-brainer. We need to use antibiotics sensibly. For those who purchase the food in supermarkets, so that's the majority of us, or in various settings, I would recommend that you boycott foodstuffs produced using antibiotics. And I imagine that there's not too many cases like that in Canada, since you banned antibiotic use in the sector some time ago. But this is not the case in the United States. It's not the case in Australia and New Zealand. And so that's rather disturbing. I think that we shouldn't invest in companies that use antibiotics in food production. And importantly, we must lobby government and the various health authorities to support R&D for AMR. So that's to say, encourage more push initiatives and also get organized so that things can be taken into long-term clinical trials, then phase 3 clinical trials, and have in place a better subscription, a better pull mechanism. Now, in the United Kingdom and also in Scandinavia, the subscription model has been implemented, and it's too early to say whether this is effective yet, but the initial signs are encouraging, and I think it would be worthwhile trying to adopt this in other geographies. And so this is the Netflix model, where the pharma company is paid up front for the product based on its value to the health system, rather than how much of the drug is actually used. So, to summarize in 4 words, there are potential solutions to the AMR problem. We can break it down into science, society, economics, and politics. I am convinced that AMR is scientifically tractable, and there's ample evidence to show that with the right organization, funding, and leadership, significant impact can be made in finding and bringing to market new antimicrobials. However, society needs to recognize the major importance of access to effective antimicrobial agents. And so changes in behavior are required, but society should also organize itself to ensure that more work is done in the AMR space. As we've pointed out during the presentation, one of the major drawbacks, the limiting factor, is in fact economics. It's the current cost the current economic models are not appropriate for the development of antimicrobials. And I think it's unrealistic to expect the private sector to bear entirely the cost of development. So this brings us to politics. It's very important that the public sector liaise with the private sector to try and come up with innovative new ways to produce the antimicrobial agents that are so sorely needed. And here I've been heartened over the years by the consistent role that the government of the United Kingdom has taken in promoting AMR awareness and in trying to innovate and find solutions. And I hope that in our very modest way, the INEOS Oxford Institute can contribute to the UK's initiative. And with that, I will stop and thank you very much for listening. And I have enjoyed giving this presentation as part of Antibiotic Awareness Week. And I hope that it has been interesting and entertaining for you. So thanks a lot.
So thank you very, very much, Dr. Cole, for this broad view of AMR and some of the specific lessons of TB. We do have time for questions. So I will invite people from the audience to come to the microphone, and you should be able to hear them. So, Marcel, I think you are first in line.
Thank you, Stuart. It's Marcel. First off, I'd never really thought of going to a meeting where TB was held up as the shining example. So thanks for reminding us that when we think things are bleak, there are actually positive lessons and messages in there. And I thought that was really valuable to hear. You described that the barriers to developing drugs and emphasized economics. Would you know, when I think of TB and the rollout of bedaquiline, the drug came out with the without a companion diagnostic. Mm-hmm. Whereas in all of medicine, when we have an antihypertensive, we have a blood pressure cuff. When we have an osteoporosis drug, we have a bone densitometer. What are the barriers to the diagnostic arm of AMR? Are they, in your opinion, higher or lower than the barriers to the drugs?
The same challenges exist in terms of. I think there are relatively simple diagnostic tools which can be developed, but unfortunately, the resistance mechanisms. There's more than one, so you would need to come up with a multiplexed diagnostic tool. Your point about bedaquiline and the lack of understanding of the resistance and that could underpin a diagnostic tool is extremely valuable. And this, I think, is a confounding factor in the diagnostic space. Often there's more than one mechanism of resistance, and the relative contribution of these mechanisms to therapeutic failure is not always clear.
Thank you, Stuart.
Hello, this is Ori Solomon. I'm a PhD student in Marcel's lab. Thank you very much for the talk. I wanted to ask you a little bit about the quote-unquote Netflix subscription model for funding, research into new antibiotics. Remember I think it was 2018-ish, when the Pasteur Act was first announced in the States and everybody got very excited. There's a lot of, it kind of lit a fire under this future for antibiotic research. And then since then, there have been a couple of countries that you had mentioned that had implemented similar models. It's also seemed to have lost a little bit of momentum. I think last year MSF came out with a report criticizing the Pasteur Act. So I was wondering what you think is the future of this model. How realistic is it for this model to be expanded beyond the countries you mentioned? And if there's any, sorry, there's...
It's just a drill.
Okay, I thought this was about my question. Which is kind of, I guess, where the question may get a little bit controversial is whether there is, you had mentioned the importance of BRICS countries or BRICS members in doing research. Is there any hope of them adopting one of these models, for example, in some of these countries?
Yeah, okay. Well, I'm going to break that down into two parts. So to answer the first part, it's one of the reasons I think why the Pasteur Act and similar such initiatives have lost momentum is due to politics. When governments change, policies change. What one government did is no longer considered appropriate. And what I alluded to with PEPFAR is a brilliant example of that. Now, the second part, whether countries like India, Indonesia, China, and so on can come up with a new way of, I mean, first of all, I think they'll certainly be able to develop drugs at a lower cost than conventional big pharma. And I don't know enough about their drug prescription systems to say whether India would be a good case setting to implement a Netflix model. I'm afraid that's beyond my area of expertise.
Thank you very much.
Do you want to step to the
Sure.
microphone?
Hi, it's Robert Platt. I'm with the School of Population and Global Health at McGill, and I appreciated both the recommendations on the drug development side and sort of individual recommendations on behavior and practices. I wanted to just maybe ask your thoughts on recommendations for public health agencies or public health practice on what they might do or we might do at a societal level for recommendations on the behavioral side, etc.
Yeah, well, I think this comes down to can be simplified by educating the public and by communicating, conveying simple, easily intelligible messages to the man in the street or the woman in the street, so that they can seize the importance of the problem and recognize that there are simple solutions which can be implemented, which will make a difference in a local setting. So I'm a big believer in public outreach and communicating to our fellow citizens.
Good afternoon. That was a great talk. This is Eric Nelson. I'm a visitor with Jesse Shapiro this week. I'm a pediatrician and a microbiologist, and I think as a thought experiment, a side opposite infection to TB might be a standard, relatively uncomplicated urinary tract infection. And so your third to last slide had kind of a wish list of things you could pull from the TB space. And I'm just curious what elements of those could be applied to our conventional approaches to treating pediatric UTI in terms of multiple agents, diagnostics, and so forth, and then the evolutionary outcomes in terms of long-term versus short-term duration therapy. Thank you.
Yeah, thank you. I'm afraid I don't have the benefit of your pediatric training, but I am aware that for instance, UTI infections are considered as should we say, a good training ground for trying to come up with new interventions to prevent, to cure the disease. And in Oxford, there's one of my colleagues, Nicole Stozer, who's leading a clinical trial doing testing a few simple new interventions to see what impact how that can improve UTI treatment. And I'm looking for, this is an ongoing trial, so we don't know yet whether it's going to be successful or not. But the premise is well based.
Anybody else?
Thank you.
To the question? Yes?
Yes, hi. My name is Michael Boucher. I'm an economist, so I can't really speak to the science here at all. But what struck me was the slide on the ingestion of antibiotics through the food chain. And I was curious how the ingestion of antibiotics through the food chain impacts AMR in humans.
Yeah, this is an important topic. It's not always as simple to explain as one might think. So obviously, the use of antibiotics in food production can have different manifestations. So in principle, the antibiotic should no longer be present in the food when it enters the human consumption chain. But that is a bit of an oversimplification, because we don't necessarily understand the impact of all the metabolites of the antibiotics which are used themselves, if you see what I mean. You can develop an assay for the antibiotic itself, but if there are a couple of metabolites which also have a knock-on effect, they may not necessarily have been discovered. I think for me, it's the massive selective pressure conveyed by the use of antibiotics in food production, which is the real scary factor, because this is what drives I think it underpins the genetic exchange of resistance determinants between pathogens which are, should we say, carbapenem resistant, and a susceptible Klebsiella living in the same environment. So you're actually creating a reservoir of resistance determinants in what we could call a naive or non-drug exposed population, which subsequently, if challenged with the appropriate antibiotic, could prove to be already resistant. And I'm fairly confident that such things take place in nature.
So with that, please join me in thanking Dr. Cole for his keynote address. And we hope that we'll see you in Montreal in a not too distant future.
Thanks a lot. Thank you. And enjoy the rest of your meeting. It looks like being a great occasion. So thank you and bye.